And thank you to the committee for organizing this session. I think it's an excellent way to showcase the things Illinois has to offer to the cancer community. So today I'd like to give you a little overview of the things we've done towards engineering single-chain T-cell receptors for cancer immunotherapy, and specifically for adoptive therapy applications. I'm going to start by giving you a brief introduction to T-cell antigen recognition and adoptive T-cell therapy, and then tell you about how we've developed these high-affinity single-chain T-cell receptor fusions. And finally, I'll show you some data on the activity of these constructs in adoptive therapy setting. So first, a brief overview of T-cell antigen recognition. T-cells are responsible for surveying the body for virally infected cells and determining whether or not cells are self or non-self. And they mediate this recognition through an alpha-beta T-cell receptor, which recognizes antigen in the form of peptide-major histocompatibility complexes. And if this recognition is of sufficient affinity, the T-cell will be activated, it will proliferate, and it will ultimately release perforins and granzymes, which will kill the target cell. So a little bit more about antigen processing. We'll take the example of a virally infected cell. Peptides from the virus are continually turned over by the proteasome, and they're presented on this protein called the major histocompatibility complex, or MHC protein. That peptide is then presented by the virally infected cell to the T cell, and recognition is mediated through this alpha-beta T cell receptor. And as I said, if recognition is of sufficient affinity, ultimately this leads to death of the target cell. So typically, the interaction between T-cell receptor and peptide-MHC has an affinity on the range of 10 to 200 micromolar, which is quite low affinity, compared to an antibody, which would be, say, nanomolar range of affinities. And I just want to point out a few features of the T-cell receptor. It consists of an alpha chain and a beta chain, and recognition is mediated by the variable domains. There are two membrane proximal constant domains, but all that's required for recognition is this variable alpha and variable beta domain. In the context of cancer, however, cancer antigens present a particularly difficult problem to the immune system, in that during development, T cells mature in the thymus, and they're exposed to all the self-peptide-MHCs in the body. So T cells against many self-antigens are deleted in this process called negative selection. And for cancer, this is a problem because many cancer antigens that are on the self surface are derived from overexpressed self proteins. So the T cells that escape negative selection have very low affinity for cancer antigens. And our solution to that approach, this problem, is to generate higher affinity T cell receptors against cancer antigens and then reintroduce those high affinity receptors back into the T cells and let the T cells target the tumor. So I want to introduce you to this idea of adoptive T-cell therapy. So as I said, this problem of the low affinity TCR we've overcome by using yeast display to engineer T-cell receptors for improved affinity. So today I'm going to talk about the 2C T-cell receptor, which has low affinity for a model tumor antigen. And Phil Holler, a former member of our lab, was able to use yeast display and fluorescence-activated cell sorting to engineer a version of the 2C T-cell receptor called M33, which has 1,000-fold higher affinity for a model tumor antigen. In the setting of adoptive therapy, the idea is that you'd be able to isolate the patient's T cells and then use a retroviral vector deliver this high-affinity T-cell receptor into those T cells. You can then expand them ex vivo and reintroduce the T cells back into the patient. And so this provides the potential for an individualized cancer therapy. Ideally, for every cancer antigen, you'd have a specific receptor, and you'd be able to type the tumor and use that specific receptor on each patient. And they've done some clinical trials at the NIH using a T-cell receptor specific for the MART1 melanoma antigen. These are melanoma metastases to the brain. And you can see before infusion of the T cells, they have these metastases. And 1 month later, after T-cell infusion, the metastases have gone away. So they've shown proof of concept that you can use a T-cell receptor in this adoptive therapy setting to eradicate tumor metastasis. However, the efficacy of these studies is limited they only saw an objective response with the MART1 TCR in 30% of patients. In addition, there's autoimmune side effects of using these specific receptors because they're choosing a self-antigen. So they saw problems with toxicity in skin, in the eye, and in the auditory system. So we think that if you could optimize the TCR, you could improve clinical outcomes by improving the affinity or by selecting an optimal target antigen. So now that I've introduced you to adoptive T-cell therapy, I want to describe our approach of using high-affinity single-chain T-cell receptor fusions. So as I said, the TCR is an alpha-beta heterodimer. And all that's required is this V-alpha and V-beta for recognition of this peptide-MHC. So we developed an approach where we made a receptor consisting of only the variable domains of the T-cell receptor fused to intracellular signaling domains. And we've termed these single-chain TCR variable fragments SCTV by analogy to the antibody single-chain FV composed of variable heavy and variable light chains. So these single-chain T-cell receptors provide some advantages. One, they can be expressed at high levels on the T-cell surface relative to the full-length T-cell receptor. Secondly, they avoid mispairing with the endogenous TCR. And there's some evidence that the mispairing between the introduced TCR and the endogenous TCR can create receptors of unknown specificity that would cause other side effects. Finally, these single-chain TCRs can mediate signaling independent of the endogenous signaling molecule. So in one protein, you have all that's required to drive T-cell activity. So using the 2C SCTV, which is low affinity, has an affinity of 30,000 nanomolar, and the M33 single-chain TCR with an affinity of 30 nanomolar, that's high affinity, we created these single-chain TCR fusions that consist of the single-chain receptor fused to a hinge and a number of intracellular signaling molecules, including the CD3 zeta subunit. We then compared these to full-length TCR constructs, that V-alpha, V-beta heterodimer I showed you earlier in the crystal structure. And as I said, the advantage of the single chain is that the CD3 subunits are built into the single chain receptor, whereas in the full-length TCR, the CD3 subunits that are endogenous are required to bring that receptor to the surface. So the surface levels of the full-length TCR are limited by the number of CD3 subunits that exist in the cell in this case. But in our case, it's totally dependent on the introduced protein. So we wanted to see if these constructs did afford advantages in surface expression and if they could mediate T-cell activity. So we introduced these cells into the 58-, T-cell hybridoma and looked at surface levels of murine V-beta 8, the V-beta used by the 2C and M33 receptor. And the full-length receptors are shown here in red and green as in comparison to the single-chain receptors, which were on the surface at 5 to 10-fold higher surface levels. So we think that this could improve the potency of the approach. Subsequently, we did an in vitro IL-2 release assay where we exposed cells transduced with single-chain TV or full-length TCR to antigen-presenting cells. And as I said, M33 is the high-affinity version. When you look at the single-chain and full-length, they released IL-2 at similar concentrations of antigen. SIY is the model tumor antigen that 2C and M33 recognize. In comparison, the low-affinity 2C receptors were not activated and did not release IL-2. So what this says is that our high-affinity single-chain and full-length TCRs have similar sensitivity. So now I'd like to show you the use of these single-chain receptors in an adoptive therapy setting. So the next two experiments I'm going to show were performed by Adam Chervin in the lab. He has a poster later this afternoon I'd encourage you to go look at. He took primary murine T cells and transduced them with either single-chain or full-length TCR. He then incubated those cells with peptide-loaded target cells that had SIY peptide on the surface, and then measured target cell lysis. So in cytotoxic CD8 T cells, the cells that are typically thought to lyse tumor cells, the full-length and single-chain TV receptors were able to specifically lyse cells expressing SIY, whereas with the null peptide OVA or no peptide, there was no killing. So this says that the M33 single chain is able to redirect T-cell mediated killing. Subsequently, Adam did the same experiment in CD4 positive T cells, which are usually thought to be the helper class of T cells that release cytokines. And surprisingly, the CD4 positive T cells were able to kill SIY targets with both the full-length and single-chain TV. So we were able to redirect another class of T cells to a model tumor antigen. So then we wanted to test out these receptors in vivo, and Carolina Soto, a graduate student in Ed Roy's lab, took a rag mouse and injected it with B16 Sergo-expressing melanoma cells. B16 is a very aggressive murine melanoma cell line. And on tumor day 5, after the tumor had been established, she injected C57 black 6 T cells that had been transduced with either single-chain T-cell receptor, with single-chain T-cell receptor into CD4 or CD8 T-cell populations, and measured tumor volume at various days. And so you can see that the M33 single-chain, CD8, and CD4 positive T cells were able to control tumor growth better than unmodified mock-transduced T cells. We were also surprised to see that the CD4 positive T cells were able to control the tumor better than the CD8 positive T cells. And we think that there may be some peripheral tolerance mechanisms that are occurring that allow the CD8 positive high-affinity T cells to lose potency over time. And so the advantage of the CAR format, or the single-chain T-cell receptor format, is that you can modify those intracellular signaling domains I glossed over earlier. So you can add other signaling components, such as 401BB is becoming more popular in chimeric antigen receptors, to try and improve potency. But this says that our single-chain approach would work with other tumor antigen. So in conclusion, single-chain TV can be expressed at high surface levels on T cells relative to full-length TCR. They avoid mispairing with endogenous alpha and beta chains. I didn't have time to show that data, but we have various lines of evidence using flow cytometry that this is the case. We also were able to show that the single-chain TV can mediate signaling through a high-affinity TCR and mediate killing. And finally, that the CD4 positive T cells control tumor growth and persist. And we think that using the single-chain approach may allow us to escape these peripheral tolerance mechanisms that, in some cases, cause the T cells to become anergic over time. And with that, I would like to thank members of the Kranz and Roy labs, past and present, specifically Adam Chervin and Carolina Soto, whose data I showed today. They also have posters at the poster session later this afternoon. And Jennifer Stone, a postdoc in our lab, she's giving a talk later this afternoon on the chimeric antigen receptor approach, where instead of using a single-chain TV, you'd use a single-chain FV derived from an antibody to target cancer antigens. I'd also like to thank our collaborators, Hans Schreiber at the University of Chicago, Phil Greenberg at the University of Washington, and Brian Baker at the University of Notre Dame, and the Medical Scholars Program for their support. And with that, I'll be happy to take any questions you might have. Thank you.
Any questions from people now? I have a question. I was wondering, I know that all the data you showed today was for the peptide SMR.
Yes.
Have you engineered any T cell yet for alternate peptides?
So we have a number of TCRs in the lab directed against human antigens. And one of those is 868, which is not specific for a cancer antigen, but this is a human high-affinity T-cell receptor that's specific for the SL9 GAG peptide. And so we introduced this 868 single-chain TCR into humans, into murine T cells, but it's a human receptor. And we've shown that it's specific for the GAG peptide and the HLA-A2-MHC. So we've extended it to HIV. In the lab, we also have receptors directed against MART1. We're working on receptors against WT1 and Survivin as other targets. Certainly. And the other thing I didn't mention is that with the full-length TCR, there is the advantage that you have CD8 helping in that interaction. So it's possible that you don't need extremely high affinities with full-length TCRs. With single-chain TCRs, it's more important. But we think that eventually, once we've elucidated all the targets, we'll have receptors against all these antigens. So it's a matter of what the best approach is to use those antigens to attack the tumor.
All right, if there's no further questions.
Thank you.